In this video, we're going to see how genes are passed on from generation to generation. We'll actually learn how scientists discover the idea that characteristics or traits are passed on in the genes of parents. We'll see what type of experiments scientists did in order to determine that. And then we're going to make some predictions about what offspring our kids might look like based on the genes that parents may have. So this is a quick little review on the terms character and traits. In this unit, we are going to be talking about mostly heritable traits, which means heritable characters, which means features that can be passed on from generation to generation. And so, for example, you might see flower color or height, or you might see shape of the peas. Are they round? Are they wrinkled? And different variations of a character is called a trait or a morph. So for example, green versus yellow peas, or you might have purple versus yellow versus white flower color. So a trait is a variation of the character. The character is the more broad term, the trait is the variant of it. I need you to understand that in organisms, traits can vary greatly from individual to individual within a population, and that's okay. That's good to have a healthy population. Many of the traits are visible to us, so I can see, for example, flower color, I can see eye color, but there are also many genetic characteristics that you don't see that are within you, the type of enzymes you might have, your blood type, for example, whether you have a certain disease or not. So those are all things that can be genetically determined, and they also count as characters. They don't necessarily have to be visible features to be heritable features or things that are passed on. So whenever you're reading your textbook, it's always a good idea to read the little learning tips that you might see in your textbook. And on the side of your textbook, you'll notice that many of the key terms that are bolded are also defined. So it's a good idea to take a look at those definitions and to maybe add them to your notes or put them into a way that you understand so that you can study and understand the concept more effectively. Again, we're talking about inherited traits, so traits that are transmitted from one generation to the next. Before we go into more details on the idea that we know and how it works today, we're going to take a look at some ideas from the past. So we already saw one idea called the performation theory, but I want to go over a few other ideas of inheritance and then how they were disproven through experiments and through observations. So again, we're going to focus on early ideas of inheritance. We already learned about this one, the performation theory, which was the idea that inside the sperm or inside the ovum, there's a little version of you, and eventually it grows into the big version of you, and that was an early idea of inheritance. Today we know that's not the case. Another early idea of inheritance is called pangenesis, which held the idea that certain organs in the female would contribute genes to the egg, and certain organs in the male would contribute genes to the sperm. So for example, the brain would give brain genes to the sperm. You might have the heart giving heart genes to the sperm. Same thing from the female. Then those would eventually combine into the egg and sperm, and then they would combine to give the zygote. So this idea was that the whole body, pan meaning the whole body, would contribute genes to the gametes, and then that would result in a zygote. Today we know that's not the case. Today we know that there's a special region, your gonads or your sex cells, in those areas, for example, that's where the sperm and egg are formed. So you might have it in the testes and the ovaries for the female, where they specialize in making the sperm and egg. So it's not all of your organs giving the genes to the sperm and the egg, it's just forming in one generalized area. Another interesting idea was the idea of the blending theory of inheritance, which basically said that you would always be some intermediate, some middle ground between your mother and your father. So if you had a tall dad and a short mom, or a tall mom and a short dad, you would be of medium height. But today we know that's not the case. There are many cases where, for example, you would have a tall dad, short mom, or a tall mom, short dad, and you can be taller than both or shorter than both. So those examples like that just shows that blending theory of inheritance is not the case. So if you're an anime fan, you might understand this idea. If blending theory of inheritance was true, well, you might look something like this if blending theory was true. So scientists essentially didn't really like this idea because there were many traits that skipped a generation. They were seen in parents but not in the kids. There were many variations of different traits worldwide. So there's no way that all those variations could be accounted for by the the blending theory of inheritance. Here's just another example to show that blending is not always the case. So if blending was true, then whenever plants with red flowers and plants with white flowers had offspring, their plants would always have, the offspring would always have pink flowers, since that's in between red and white. But that's not always the case. Sometimes in nature we see this, where you have red flower plants, white flower plants, and they give more red flower plants. You might also see this, where you have red flower plants and white flower plants, and they give more white flower plants. So these are all examples that go against that blending theory of inheritance. So one of the most prominent scientists that came up with an idea that actually we continue to use today is Gregor Mendel. He came up with a hypothesis called the particulate hypothesis of inheritance. And this is what we know today as the gene idea. So his words were, parents pass on discrete heritable factors that retain their separate identities in offspring. He was basically saying that parents pass genes on to kids, and then those genes stay the same in the offspring. So your mom might pass on a gene for brown eyes, your dad might pass on a gene for blue eyes. They don't blend together in any sense. There's no such thing as a little mini version of you inside the sperm or the egg. And he came up with this idea. Now, he didn't use the word genes back then, but today those heritable factors, we refer to them as genes. Now, how did he figure this out? Well, he did a bunch of scientific experiments to determine this idea. And so the rest of this PowerPoint will be focusing on the experiments that Gregor Mendel did in order to come up with this idea of the particulate theory of inheritance. He observed patterns of inheritance. So basically he bred many different organisms, particularly pea plants, and then noticed many patterns in the kids that were made, and based on the patterns that he observed, he was able to come up with this idea of inheritance. So he started to see patterns, he started to make guesses, hypotheses, and he said if his hypothesis is correct, then when two parents with these characteristics have kids, the kids should look like this. And he found that many times his hypothesis was supported by the genetic experiments he was carrying out. So we often refer to Gregor Mendel as the father of genetics. He came up with the idea of genes being passed on. Again, he didn't use the term genes because we didn't really know about DNA and chromosomes back then, but he came up with the idea of heritable factors being passed on from generation to generation. So when you do your readings, make sure you read up on Gregor Mendel a bit to find out more of his background, what he did for a living, what he studied, and why he was interested in genetics. So many scientists were studying genetics back in the day with Gregor Mendel, but what really set Mendel apart was that he had a really good experimental design. They were simple experiments, but they were powerful. And what made his experiments really powerful are the following things. He had a lot of repetitions. He didn't just do one experiment one time and say that this was the new rule based on one experiment. He repeated the experiments over and over and over again so he could make generalizations about the patterns that he noticed. He kept very good records. He made very good observations, and making those observations and keeping records of those observations allowed him to notice patterns in the results that he was getting. He took many types of observations, some quantitative ones and qualitative ones, so he didn't just say, I'm getting some red flowers and some white flowers. He actually counted how many of each type he was getting. So he really did a good job at analyzing the numerical data in order to find trends and patterns and then to come up with a general idea about inheritance in the plants or organisms that he was studying. So in order to study genetics, Mendel decided to use the pea plant. So he was using peas. And there's a reason for that, or a few reasons for that. First of all, they were available in Europe. They were commercially available in Europe, so very easy for him to get his hands on. They're available in many different varieties, so he could maybe test out breeding different varieties of peas. So it gives him some choice in terms of the experiments he can do. Peas also have a very short generation time. Basically, they reproduce very quickly. They make babies quickly, so he could study this reproduction in a timely manner. He didn't have to wait years to see what traits were developed. And also another thing that was particularly useful with using peas is that Mendel could control their mating. He couldn't really take animals or humans and force them to mate and see their characteristics. So Mendel could actually do cross-pollination. He could take some of the sex cells from the plant with a little brush and then put it onto another plant with that brush. So he could do cross-pollination in order to control the mating of the plants in a specific way. He might say, I want to mate this plant with the purple flowers with that plant over there with the white flowers. So he'll take his brush, take the pollen from the purple flowers, put it onto the flowers put it onto the plant with the white flowers, and then see what happens at the end of the day. So these are the main reasons why Mendel decided to use pea plants to study genetics. There's another reason as well, and that other reason is that peas have many dichotomous traits. Another way to think of it is contrasting traits. Sometimes people call that either or traits, meaning they're very, very easy to count and tell apart. For example, the flower is either purple or white. The flower position is either axial or terminal. The seed color is either yellow or green. There's no kind of in between. It's really easy to spot. Okay, there's 5 green ones, 2 yellow ones. Very straightforward. So those contrasting traits made it very easy for Mendel to count and to keep accurate records. Whereas if there were a lot of in-between traits, it might have been more difficult to count and taken them less time. So it was a lot less ambiguous with these dichotomous. Dichotomy means basically either or. It's black or it's white. So round or wrinkled. Over here you can see some of his numbers and his ratios that he figured out, and we'll talk more about these later on. But the main reasons for which Mendel chose peas were just discussed. Make sure you understand that very clearly. So where did Mendel start? Essentially, he started off with what's called true breeding plants. That was his first generation of plants, true breeding. That means they were plants that were only ever bred with other of the same types of plants. So for example, he would have taken, let's say, some true breeding plants with purple flowers. Those true breeding plants with purple flowers are only ever bred with other purple flower plants. So they only really had the purple flower gene in them, nothing else. And so he called that the pea generation. P for parental, not for pea plant, but for parental. And essentially he just did some self-pollination. He took pollen from a purple flower plant and then added that to the female part of the plant and then saw the babies that were produced. So plants can actually reproduce within the same plant. They have both male and female parts. So for example, over here, there would have been some of the male gamete, and that was added to the female gamete over here, and then that made the baby, the zygote, which would develop into other plants. But the idea is that his first generation is called the pea generation, and they were true breeding plants. They were only ever bred with plants of the same variety. So these were true breeding purple plants, or there may have been true breeding white flower plants. So it's called true breeding. Sometimes you can call them purebred as well. And again, that was a pea generation. And what he noticed when he did this self-pollination is that whenever you bred purple with purple, all the kids ended up being purple. Whenever you bred pure breeding white with pure breeding white, all the kids ended up being white. Whenever you had let's say pure breeding green peas and pure breeding with pure breeding green peas, then all the kids ended up being green. That was his first general observation that he made about doing mating between pure breeding individuals of the same variety. So again, that was called his pea generation. Now, what he did is he decided to try something a little bit different. He decided to do what's called a monohybrid cross. A monohybrid cross is when he took a pure breeding plant of one variety, so let's say pure breeding tall plants, so plants that had only ever been bred with other tall plants, and he crossed them, that X means crossed them or mated them, with pure breeding short plants. Pure breeding short plants. So that means they were short plants that had only been bred with other pure breeding short plants. And those would produce kids or offspring. So these pure breeding plants, they were the P generation. He mated them and they had kids. And those kids, he called them the F1 generation. The first filial generation. And those plants were hybrids. The reason they were called hybrid plants is because now they had a mixture of, we're going to use the word gene here, he didn't use the word gene, but they had a mixture of genes. They had tall genes and short genes. They were no longer pure breeding anymore. The tall was not only bred with tall, the short was not only bred with short anymore, the tall and the short had been bred together, so they were called hybrids. So a hybrid is just basically a mix of different pure breeding plants. Now we call this a monohybrid cross because we're just really keeping track of one particular trait here. We're keeping track of height right now. So we did a short pure breeding pea plant with a tall pure breeding pea plant. So I call that a monohybrid cross. If I was studying two characteristics at once, I might call that a dihybrid cross. But right now we're just dealing with one trait or one characteristic, the short version of the trait and the tall version of that trait, of that characteristic. And so now we're going to take a look at the results he got. So again, we have our pure breeding tall plant and our pure breeding short plant. Whenever you cross two pure breeding organisms, two different ones, we call that hybridization because you're creating a hybrid, a mixture of the two. Now I just want to show you a little notation here. Our pure breeding tall plant, we're going to call it big T, big T. That means that it has two tall version of the genes within it. We are studying plants here. These particular plants, these pea plants, are diploid. That means they have two versions of each gene. A pure breeding tall plant is going to have two tall version of the genes because it was only ever bred with tall plants. The pure breeding short plants is going to have two short versions of the height gene. So we'll call that little t, little t. So tall is going to be big T, big T. Short is going to be little t, little t. So we crossed the pure breeding tall with the pure breeding short, and his results were not this. He was trying to see if he would get this because a lot of people thought that there would be a blending theory of inheritance happening, but he didn't get medium plants. Because he didn't get medium plants, he knew that blending theory of inheritance was not necessarily being supported by this experiment. But this is what he did get. He noticed that when he crossed the pure breeding tall with the pure breeding short, all plants ended up being tall. They were the same height as the tall parent. So it wasn't blending. He thought that something interesting was happening. He noticed, okay, there was no more tall trait here. The tall trait of the height character seemed to have disappeared. Now what he didn't know is that the gene the short trait seemed to have disappeared. Let me go backtrack for a minute. So again, pure breeding tall, pure breeding short, all the offspring ended up being tall. The short trait of the height character seemed to have disappeared. He didn't know where it went. Okay. Now what he didn't know is that short version of the gene is still hidden in there somewhere, just not showing up. So what he created here was the F1 generation. They're the hybrid offspring, meaning they're no longer pure breeding because they had been created by mixing tall pure breeding and short pure breeding. So we're going to give this a two-letter notation. We're going to call it big T, little t. Big T, little t. As a tall version and the short version of the gene, of the height gene. Okay. Now Mendel didn't know this at the time, but we do. Okay, it's going to be our secret for now. Now, he then decided to take the F1 generation and mate them together. So he was going to take genes from an F1 plant and mate it with another F1 plant. So again, just discussing his results here for his first experiment, P generation, all pure breeding parents. Mixing them together, you get all tall plants. The short one seemed to have disappeared. The short trait disappeared for some reason. We don't know why. So right now, we have 100% tall. 100% tall. And 0% short. Now I want you to keep in mind that even though we're only studying 4 plants here, Mendel didn't just study 4 plants. There were a lot more plants, hundreds, thousands of them. But we're just simplifying it for the sake of this. So we're simplifying it for the sake of our presentation to a manageable number, but he studied a lot more than just 4. Okay, there were many repetitions that we simplified in the textbooks. And this pattern is something that he noticed for many characters, not just for height. So he also went on to study the flower color. He noticed when he bred purple flowers with white flowers, all the offspring had purple flowers. So that white flower trait had seemed to disappear. There was 100% purple. He noticed it with the pea colors. He noticed it with the shape of the peas, the texture of the peas, with a variety of different things that for some reason, whenever he crossed some pure breeding parents in the pea generation and made hybrids, one of the traits seemed to have disappeared every single time. The short, the white, so on and so forth. And so here you can see the pollination event that Mendel did. He took some pollen from the male part of a purple flower and put it into the female part of a white flower. He then took a seed that was made from that, the peas or the seeds there, planted that, and the plants grew. And he noticed that when the plants grew, they all had purple flowers, and there were no longer any plants with white flowers. So the white flower had disappeared from there. He called these hybrids, though, because they were now a mixture of 2 different pure breeding plants, which were our parental generation over here. This was our P generation. This is our F1 generation over here. So that was Mendel's first experiment. Mendel wasn't done yet. He then decided to take the F1 generation and cross them together. So he took an F1 plant, big T little t, crossed it with itself or maybe another F1 plant, big T little t, and he then analyzed the results and saw something quite interesting. This is a summary of the results, but basically the idea was that the trait that had disappeared, so we're going back to our height trait here, our height character and our tall trait, the trait that had disappeared had actually returned. He noticed that in the generation produced by the F1, so this is our F1 over here, first filial generation, they had kids and they were called the F2, the second filial generation. In the F2 generation, 3/4 of the plants were tall and 1/4 short. So we call that a ratio. We call that a 3-to-1 ratio. That is today called the Mendelian ratio. 3 of the one that had taken over to 1 of the one that had disappeared. So again, that trait that had disappeared had returned. So the conclusion that Mendel came up with was that in the F1 generation, the trait for the one that disappeared was probably still hidden within. It was maybe masked by the other trait, by the tall trait. But then once he mated the F1 generation, somehow it became unmasked. It reappeared again. So it was never really gone. It was just hidden by one of the other traits. So we're going to go take a look at that experiment in a bit more detail with a few more visuals to understand the concept. So here's our second experiment. He took plants from the F1 generation. These are our hybrids, the F1 hybrids, big T, little t, big T, little t, and he crossed them together, and he got the F2 generation, second filial generation. And this is a ratio he got. So again, he didn't study just 4 plants, he studied many plants. And if he simplifies the ratio, when I say simplify the ratio, I mean, for example, let's say I have 4 out of 8, and I simplify the ratio, that's the same thing as 1/2. So he studied all these plants, and he got 3 of the plants were tall, and 1 was short. And he saw that with many characteristics. He always got this 3-to-1 ratio. 3 of the one that took over in the first generation to 1 that was hidden in that F1 generation, F1 filial generation offspring batch for many of the characters that he saw. So this one that had disappeared had returned. I just want to go over the letter notation here. You'll notice that they do have some slightly different letter notations. You have some of the tall plants big T, some of the tall plants big T, little t, and our short plant that was back to little t, and we'll go over why that is in just a bit. So again, Mendel saw this ratio for many characters, so he then took the F1 generation, for example, from the purple flowers, crossed them together, and he got 3-to-1 ratio here. So you'll notice 705 purple flowers to 224 white flowers. If you simplify that, that's a 3-to-1 ratio. It basically means 75% of them, of the plants, are the one that took over in the F1 generation, and 25% of the plants are the one that had disappeared. But we know now they hadn't really disappeared. They were probably just hidden or masked by the other character trait in the F1 generation. And so you can see that over here for many of the characteristics he studied, 705 to 224, that's about a 3-to-1 ratio. For the flower position, 651 to 207, that's again a 3-to-1 ratio. It's, it's not exactly 3-to-1 because making babies and their characteristics are always up to chance, but it's really, really close every single time. Yellow to green seed color, 6,022 to 2,001, that's about a 3-to-1 ratio. And then round to wrinkled, 5,474 to 1,850, that's again close to a 3-to-1 ratio. And again, this is our Mendelian ratio. Whenever you cross 2 hybrids, so for example, big T, little t times big T, little t, you're always going to get this 3-to-1 ratio. 3 of the trait that took over to 1 of the trait that had disappeared, that was gone in the F1 generation. So again, you can see all that here with these characteristics that you studied. Very, very close, all of them to a 3-to-1 ratio. That ratio is going to be very important for you to remember when we do practice problems in crossing individuals to see what type of babies they'll produce. So we're going to go over Mendel's main findings and conclusions based off of the experiments that we just discussed. So even though he didn't know about genes, he still discovered all these ideas. He wouldn't have used the word genes, he would have used other words, but we're going to now replace those other words with things that we know today. Genes and alleles, and we're going to talk about chromosomes a little bit, but these are, in general, the main ideas that we came up with due to Mendel's findings with those simple but powerful experiments. So we're going to go over each of these one at a time in the next few slides, along with some examples. It's very important that you understand meiosis because part of his findings does relate to meiosis. We're now combining meiosis to Mendel's work. So the first point we're going to talk about is that alternative versions of genes exist. The alternative versions of genes are called alleles. And in diploid individuals, we inherit 2 alleles per character, one from mom, one from dad. So let's just take a look at this example over here. Here we have some homologous chromosomes, and we know homologous chromosomes have the same length, same centromere position, and same gene loci, so same position of genes on a chromosome. They would also have the same genes, but they could have different versions of the genes. So for example, in the plant that we saw that was a hybrid, for flower color, it might have had purple flower on one homologous chromosome and white flowers on the other. So maybe it got the purple flowers from mom and the white flower one from dad. So one of the alleles is represented by an uppercase letter and one is represented by a lowercase letter. So for example, I might represent the purple one with uppercase A and the yellow and the white one with lowercase a. Or it doesn't really matter, I could have done uppercase P and lowercase p, right? Uppercase P for purple and lowercase p for white. So for the tall plant, maybe there was a tall gene on one chromosome, or tall allele I should say on one chromosome, and a short allele on the other. So they're the same gene, just there might be some slight variation in their nucleotide codes, and that's the reason why one of them might code for purple, whereas the other might code for white. And so Mendel was able to figure this out from his experiments, that there's alternative versions of these heritable factors or these genes that exist, and for diploid organisms, we inherit 2 alleles for each character. So that was the second point. For each gene, an organism will inherit 2 alleles. So like I said, you might get the purple from mom and the white from dad. Those alleles are found on the homologous chromosomes. They're at the same general location on each chromosome, the same relative location. And again, the location for a gene is called the locus. So we have the purple and the white. Now, you don't always have to get 2 different versions of the gene or 2 different alleles. Sometimes you can get 2 the same one. So we saw, for example, the short plant had little t, little t. So on one chromosome it would have little t, on the other it would have little t. It inherited the same version from both mom and dad. Or some of them might have little p, little p. So 2 white ones, one from mom, one from dad. If I were to represent this organism's genes for flower color, I would write it like this. I would put the uppercase one first, followed by the lowercase one over here. So if I write this, someone would know that on one chromosome there's the big P, on the other chromosome there's little p. Now, there's a term for what versions you inherit. If you inherit 2 different versions of a gene, so let's say big P, little p, so big P, little p, then you're called a heterozygous individual. Heterozygous. Hetero meaning different. So this individual here is heterozygous for flower color. Now let's say you inherit the same version of the gene, the same alleles for a character. Let's say big P on one chromosome and big P on the other. In that case, I'm called homozygous, where you have 2 of the same alleles. So it could be big P, or it could be little p, little p. The main point is that it has to be 2 versions of the same allele. 2 of the same versions of the allele, rather. So again, here we have an example of heterozygous versus homozygous. Remember that on a chromosome you might have different genes. So for some genes you might be heterozygous, for other genes you might be homozygous. So over here we have the MD gene. You can see we have little md, little md. Sometimes you can represent the gene by 2 letters. Doesn't really matter. This could have been little a, little a. But the point is that we have the same version of the gene, 2 of the same alleles for MD, so this would be homozygous. Whereas over here we have the Li plus, and over here we have the little Li. So over here for this particular gene we are heterozygous. So you can be heterozygous for some genes, homozygous for other genes. Now the third point that Mendel came up with is that if you do inherit 2 different alleles, then usually one is going to be dominant and one is going to be recessive. Essentially, one will take over, one will be represented or expressed, that means shows or gives the appearance, and one will be hidden if they're inherited together. So that one's called the recessive one. And so in Mendel's experiments, where he took the true breeding plants and mixed them together, he noticed that one disappeared. The one that disappeared was the one that was recessive. So in this case, let's say this is our dominant allele, big T, and this is our recessive allele, little t. The reason he knew that tall was dominant is because it masked or hid the short one. So in this example here, we have a heterozygous individual where big T, the tall allele, is dominant to the short allele, the recessive one there. So recessive is always shown by a lowercase, whereas dominant is always shown by an uppercase letter. And the reason and the way we know if something is dominant or recessive is by taking a look at the patterns of inheritance, so how things are inherited. And we notice in this case that the short one disappeared, so it's the one that must be the recessive, and the tall must be the dominant allele. So if we take a look at this example here, we have the purple flower, pure breeding, mating with the white flower, pure breeding, and all are purple, so the white disappeared. So we have big P, little p. That means that our white is going to be our little p. And we know that because the white disappeared, whereas the purple took over. So purple is our dominant allele, and white is our recessive allele. So if I give you similar experiments that Mendel conducted, you should be able to figure out, based on results like this, which one is dominant, which one is recessive. And so this idea of dominant versus recessive, Mendel called it the principle of dominance. So when an individual has two versions or two alleles, different alleles, only one is expressed, and the one that's expressed is a dominant one. The one that's not expressed is the recessive one. So I'm just going to go over Mendel's experiments, this time looking at these letters in more detail. So for the pure breeding has, they're homozygous for their characteristics. So they have two of the same alleles. So tall had big T, short had little t. This one is homozygous for big T. This one is homozygous for little t. So we would call this tall one here with the big T homozygous dominant, and we would call this one with little t homozygous recessive, meaning it has two recessive alleles. Shouldn't say genes, should say allele. And this one here has two dominant alleles. So homozygous dominant, homozygous recessive. And they produced hybrids, individuals with two different alleles. Those hybrids, we would now more correctly call them heterozygous individuals. And we can tell that the big T, the tall, was dominant because the short one disappeared. And of course, this is a lame joke, or not. It's probably the most truthful thing you'll hear all year long. So just a summary of the terms we learned: homozygous, having two pairs of identical, having a pair of identical alleles, and those were the pure breeding plants that Mendel had discussed. And again, this would be a homozygous dominant, and this would be a homozygous recessive over here, two recessive alleles, two dominant alleles for a particular character. And here we have our hybrids, which is our heterozygous individual. And so you can see that illustrated here, and that applies to all of the other characters that Mendel studied as well. Now, let's analyze these two characteristics. Just by looking at these two characteristics, which trait would be dominant? Having an extra digit or not having an extra digit? How can you tell based on this picture? Which is a dominant trait? Polydactyly, meaning having the extra digit, poly, many, or let's call it the normal, having the normal number of digits. You can pause the video, think about it, then commit to an answer. Choose either normal or polydactyly for being dominant. Now, if you said normal, I understand where you're coming from. Most people think that if something is better or looks better, that's going to be dominant, but it's not always the case. You can have diseases that are dominant alleles, Dominant doesn't mean the one that is the better one for you to have. Dominant doesn't mean that it's the one that's more common in the population either. In fact, the normal is actually the recessive one, whereas the polydactyly is actually the dominant allele. Yet, normal number of fingers is more common in the population, whereas more fingers, polydactyly, on one hand, is less common in the population. So dominance does not refer to how common something is in the population or if something is good or bad. It just refers to whether or not it would mask something. So for example, if an individual with polydactyly had a baby and it passed on the, let's call it big D gene, and this one over here passed on the little d gene, then the baby would have big D little d, and as a result would have polydactyly. But it's not as common in the population. So dominance does not refer to how common something is in the population. It just refers to whether it's going to be the one being expressed or not in the individual. So that's another learning tip you should pay attention to in your textbook as you're reading. It does not mean the better allele or the more common allele. That is a misconception. So we have two important terms to discuss. We're going to be using these terms quite a bit in this half of the genetics unit. Genotype and phenotype. Genotype, think gene, refers to your genetic composition. Which alleles do you have? So if I were to say, tell me the genotype of this plant over here, you would tell me it's big T, big T. If I were to say, tell me the genotype of this plant over here in the middle, you would say big T, little t. You'd write down like that, big T, little t. And of this, the genotype of this plant here, you'd say little t, little t. So you're looking at the genetic or allelic composition of the organism. The phenotype, on the other hand, refers to you describing the appearance or the expression of the characteristic. Now, to simplify, we sometimes say the physical appearance, but it could also be other things that you don't see, but it's not the genetic composition. So for example, the phenotype of this plant here, a big T, the phenotype is that it's tall. The phenotype of this plant over here, the big T, little t, is that it's tall. So they both have the same phenotype, the tall phenotype. Whereas this one here, little t, its phenotype is that it's short. So genotype, the letters to represent what genes or alleles they have, rather. And then phenotype, think physical, pheno, the characteristics that's expressed. So can you pollinate or can you not pollinate? And that might be determined by genes. And if you were to describe the genotype, you would tell me what genes they have to be able to pollinate or not pollinate. So one of our last rules that Mendel, or conclusions that Mendel was able to come up with, is this idea called the law of segregation. We learned that during meiosis that homologous chromosomes separate. But with that comes a big implication. Let's say you had a parent over here. Heterozygous parent, big A, little a. It goes through meiosis and it produces gametes. What I want you to notice is that the alleles separate during meiosis because the alleles are on homologous chromosomes, and homologous chromosomes separate during meiosis. So here I have big A, here I have little a. They're going to separate. So in one gamete you'll get big A, in the other gamete you'll get little a. I'm just looking at one of the one of the daughter cells dividing to make gametes. That's why I don't have 4 here. So essentially you have a 50% chance of getting big A and a 50% chance of getting little a. So during gamete formation, the alleles will separate, and that's because they're on homologous chromosomes. So if a parent was heterozygous, it has a 50% chance of passing along the dominant allele and a 50% chance of passing along the recessive allele. So we're going to take a look at this in more detail, studying meiosis and combining it in more depth. So here we can see an individual that is heterozygous, big G, little g. We always represent it with two letters only. I know there's 4 letters here, but that's because the chromosomes are replicated. So big G, little g. Notice that big G and little g have separated. They segregated. They're in different gametes now. You'll have 2 of the 4 gametes produced are big G, 2 of the 4 gametes produced are little g. So when you represent the genotype of the individual of the parent, you have 2 letters. For the gamete, you use 1 letter. You have a big G gamete, because gametes are haploid, right? This is diploid, 2 copies of the gene, 2 alleles, and the gamete is haploid, so 1 copy of the gene, 1 allele. So basically 50% chance of getting big G, 50% chance of getting little g. That's the chance they have to pass on, 1 allele over the other. Now here we have a homozygous individual, homozygous dominant, big G, big G. And that's right, you guessed it, it only has big G to give, so all its gametes are going to be big G. The alleles are still going to separate, it's just that you still have big G here and big G here. So we have big G, big G, big G, big G. So all the gametes are big G. That's all you can pass on if you're homozygous dominant. And you're probably getting the idea that if you're homozygous recessive, that you'll only pass on the recessive allele. So just take a look at what that looks like in terms of meiosis and fertilization. Let's say you had a heterozygous parent over here, and that heterozygous parent goes through meiosis. 50% of its genes, of its gametes would have big T, and 50% will have little t. Here's the other parent, parent 2, also heterozygous. 50% big T, 50% little t. And that gives you a lot of combinations. So for example, this gamete might fertilize with that gamete, and you'd get a big T, big T. This one here might fertilize with this one, and you get a big T, little t. This one here might fertilize with this one here, and you get a little t, little t. So there's different variety of gametes, and you get different combinations based on what gametes are selected to go through the fertilization process. Now in this case here, we had our, this is similar to our F1 generation being crossed to give F2 generation, because it was heterozygous crossed to give your tall individuals and your short individual that had come back. So this is a summary of the experiments that we saw. Pure breeding individuals were crossed together. So let's say we had big T, and then little t. So homozygous recessive with homozygous dominant. They gave a F1 generation that was a hybrid. The short trait had disappeared, but it was still hidden within, so we have our heterozygous individual here. We allowed the F1 generation to self-fertilize, and so basically it was just crossing F1 with F1, so it would kind of look like this, big T, little t, cross with big T, little t, and the end result is that we got our 3-to-1 ratio. Whenever you cross 2 heterozygous individuals, you get a 3-to-1 ratio. Whenever you cross 2 homozygous individuals, you're going to get 100% of something. But let's just go over this in more detail. So if we were to study our Mendelian ratio, we would get 3 tall, so 3 dominant ones, to 1 recessive one, to 1 short one. Now this is our phenotypic ratio. We're looking at the phenotype. In reality, you had, or at the genotypic level, you had one of the tall plants was big T, 2 of the tall plants were big T, little t, and then the short plant was little t. It had no choice to be little t because if there's a big T with it, it would be tall. Okay, so phenotypically speaking, we have 3 tall to 1 short after you cross hybrids with each other to get the F2 generation. And it's these experiments, this set of experiments that allowed Mendel to come up with those ideas that we discussed in the previous slides. Again, here's our summary of everything Mendel came up with. You should read this summary slide and then go back and study each one of the slides with more detail to make sure you understand the principle of dominance, the law of segregation, and what an allele really means. You should also be able to focus on the idea of writing down the genotype, for example, big T, big T, and knowing how to name the individuals. Are they heterozygous? Are they homozygous dominant, homozygous recessive? And then knowing what type of gametes could be made. So for example, this individual here could make a big T gamete or a little t gamete. It can make both gametes. 50% of the time it'll be big T, 50% of the time it'll be little t. This individual can only make big T gametes because there's only big T to give. And this individual can only make little t because there's only little t to give. Gamete genotypes are always one letter because they're haploid. The parent genotypes or the individual genotypes are always going to be two letters because they're diploid. So we'll do a few sample problems together. So in this case, we have a scenario, polydactyly allele, big D is dominant to the normal allele, little d. Based on information, what is your phenotype? So your phenotype, if you were to study it, you'd have to tell me, are you a polydactyl, or are you an individual with a normal number of fingers on one hand? And so for me, I would say that I have 5 fingers on each hand, so I have the normal phenotype. For genotype, you'll notice that there might be 2 choices, and you could actually narrow it down if you knew what your parents were, but there's 2 possibilities. So actually no, for normal there's not 2 possibilities, there's only 1 possibility. For normal, for me it's little d, little d. I have 2 copies of the recessive allele, so I'm homozygous recessive. But for a person with polydactyly, you'll actually see there's 2 possibilities. That person could be big D, or big D, little d. It all depends on what their parents had. Think about it. If their parents were both big D individuals, then the baby would have to be big D, because the baby could only get big D. But if at least one of the parents had big D, little d, then the baby might have big D, little d as well. We don't know, because one of the parents could have given a little d in there. So you need more information to accurately determine which genotype they have, but you should know that there's at least 2 possibilities for polydactyly. You should be able to do practice problems like this, so you should be able to pause the video, try it out yourself, and then check the answers after. So we have a scenario again. Brown allele is dominant to blue allele for eye color. Determine the phenotype of a person with the following genotypes. So we have big B, little b. We told you that the brown allele is dominant, so this person here, their phenotype would be brown eyes, because it's dominant to the blue eye. Big B, homozygous dominant, also brown eyes. Little b, that's the only way that you can see the recessive, blue eyes. And then here you'd have to classify the individuals as being homozygous dominant, homozygous recessive, or heterozygous. Remember, hetero means 2 different alleles, so this is our heterozygous one. Homozygous dominant, 2 of the dominant alleles, right there. Homozygous recessive, 2 of the recessive alleles, right there. You should be able to try this one out yourself, and then check your answers after. So the allele for coolness, C, is dominant to the allele for uncoolness, little c. Determine the genotypes for the individuals who are cool. There's 2 possible ones, big C, little c, or big C, big C. Determine the genotype for individuals who are uncool. Well, there's only one, since that's recessive, and that's little c, little c. In humans, detached earlobes are dominant to attached earlobes. Write the homozygous dominant genotype. Big D. Write the homozygous recessive genotype. Little d, little d. Write the heterozygous genotype. Big D, little d. Genotype for an individual with attached earlobes. So attached. Attached is recessive, no choice but to be little d, little d. Genotype for an individual with detached earlobes. That's dominant, but, so that means there's 2 possibilities, big D, little d, or big D, big D. Can you find the phenotypes if I give you the genotypes? So again, you should be pausing this video and trying it yourself and then checking your answer. So if you're big D, you have that dominant allele, so you would be detached. If you're big D, little d, also detached. I'm just going to put this here to show it's the same. And if you're little d, well, you would be attached earlobes. Same idea. In humans, ability to roll tongue is dominant to not rolling tongue. So write the following, pause the video, check your answers. Homozygous dominant genotype, big R. Homozygous recessive, little r. Heterozygous, individual who cannot roll tongue, it's recessive, so little r. Individual who can roll tongue, big R, little r, or big R, big R. To narrow it down, you'd have to know what your parents are, or what type of kids you might have with somebody else with a certain characteristic. The phenotype for a person with big R, big R, you can roll. Big R, little r, you can roll because it's dominant. Little r, not roll. So no matter how hard you practice, you won't be able to roll your tongue if you have little r, since it's genetic. Those genes determine what proteins you have, which determine maybe the muscles in your tongue to allow you to coordinate how you roll it. Here's another example. In plants, red flower color is dominant to white flower color. Based on this information, write the phenotypes and genotypes in the pictures below. So we have our red flower color. That's our phenotype, red. Our white flower color, that's our phenotype, white. The expression of the trait, the way it looks. Our genotype for red, well, red is dominant. So we know our genotype could be big R, little r, or big R, big R. For white, it's recessive. So it has no choice but to be little r, little r. If I don't give you a letter for the alleles, you can make up your own letter. Gametes that can be made. Well, for the white one, it's only little r. For the red one, if you're big R, the only gamete you can make, all gametes that you make, let's say for one round of division, would be big R, 4 of them. For the gamete for the red flower, that's heterozygous, it could make big R or little r. Here are the learning goals for this PowerPoint. You should be able to do the majority of these without much difficulty. Go through the PowerPoint in some more detail. Go to the readings in your textbook to understand the experiments that Mendel did. It'll be very important for you to understand how to use this notation for homozygous, heterozygous, homozygous dominant, recessive, so on and so forth, to understand the terms genotypes and phenotypes, because we're going to be doing a lot of practice problems and crosses to predict phenotypes and genotypes and ratios like Mendel saw. Because those type of experiments are still very useful today to predict patterns and to predict what type of offspring you'd produce. You have some more practice problems to do over here with different characteristics. If ever I give you a scenario and there's no picture associated with it, in this case there is, it might be a good idea for you to maybe look up a picture of what that characteristic is so you understand what it looks like phenotypically speaking. Over here we can once again see our father of genetics, Mendel, and this is a Halloween costume that I came up with where I was a zombie version of Mendel, and later on you'll understand what this image is here on my apron with the Zeds over here and this little grid with all of them in there. And instead of blood, you'll notice the creative peas and the leaves in my hair. So to finish off, I want to show you an animation of meiosis. You've actually already seen this animation in the meiosis presentation that you studied in the first half of genetics, but this time what we're going to do is we're going to add the alleles to this. We're going to take a look at how the alleles come into play and that law of segregation in more detail. So right now we have our interphase happening. Here we can see our prophase essentially, and we can see we have a heterozygous individual, big T, little t. Again, I know you see 4 letters, but that's because of that DNA replication. So we're in metaphase now. As we go through anaphase and telophase, the big T and little t will separate. So we're going to have our law of segregation there. And so you can see the gametes produced are 50% big T and 50% little t. That's why a heterozygous individual can produce more than one type of offspring depending on who it's mating with. The readings for this section are included in pages 181 to 188. Make sure you complete the learning goals and the worksheets and other activities that we provide you with in order to make sure you understand the information well.